Imagine this. You're baking. Then you accidentally pour a bit too much lemon juice. Then suddenly, the whole thing is sour. Or imagine your body. If it becomes too acidic, you could literally get sick or even die. So thankfully, chemistry has something that prevents these wild changes. We call it buffers. Hi everyone! My name is Robb C. Suaybagio, together with my partner Angel Marie de Gracia. We will be discussing the history and importance of buffers. First and foremost, what exactly is a buffer? A buffer is a solution, in other words, a liquid mixture of two or more substances that resist changes in pH when small amounts of an acid or base are added. Pero unsa man ang pH? pH stands for potential of hydrogen. pH tells you kung acidic ba or basic ang isa ka solution. It goes from 0 to 14. 0 to 6 means acidic. For example, lemon juice or suka. And 7 is neutral, like pure water. And 8 to 14 means basic, like sabon or toothpaste. It helps maintain a stable chemical environment, crucial for many biological and chemical reactions. So basically, buffers are like peacekeepers of chemistry. Gina-stop niya ang acids ug ang bases from causing chaos. Gina-keep niya ang tanan balanced and stable. Pero how? A buffer usually contains a weak acid and its conjugate base, or a weak base and its conjugate acid. But what does that mean? An acid means a substance that increases hydrogen ions in a solution, or it makes it more acidic from the name itself. And the base is a substance that reduces hydrogen ions or increases hydroxide ions. Now, what is a weak acid? Acid siya. It is an acid that doesn't completely break apart in water. For example, acetic acid. Acetic acid, kay makita na nimo siya sa suka. And its conjugate base, it is called acetate. Muna siya ang leftover part after the acid gives away one hydrogen atom. The two of them, acid and base, work together like a team. Kay, kung naay masobraan sa isa, kay neutralize na sa isa. If masobraan ang acid, neutralize na sa base, and vice versa. So that is how the pH stays stable. Buffers are essential in living organisms, chemical reactions, and industrial processes. Buffers are important because they maintain a stable pH level in various systems. pH stability is crucial since even small changes in acidity or basicity can affect chemical reactions, biological processes, and product quality. So, ganun gamatter mo na? Because most chemical reactions only work properly at a specific pH. For example, enzymes nato, ang proteins that help reactions happen faster in our bodies. They can only work at the right pH. So, if the pH suddenly changes, these enzymes stop working. And that is why our body has natural buffers. Ay tawag ana, these are called the bicarbonate buffer system in the blood. If our blood gets too acidic, the bicarbonate absorbs the excess acid. And if it gets too basic, it releases acid to balance it again. So, let us move on to the importance of buffers. Buffers, kay dili lang na siya importante sa atong body, but they are used and can be seen everywhere. Without buffers, even a small addition of acid or base could cause a large change in pH. In biology, ang buffers ay gina-keep niya ang body fluids stable, pareha sa blood like I said earlier, saliva sa tunglawan and even inside cells. If change ang pH, damage ang enzymes, and life processes will stop. Buffers ensure reliable results in laboratories and consistency in industrial products. In laboratories, buffers are used to maintain the right condition for experiments. Sa industry's part, buffers are used in literally everything. For example, our food, to keep flavor, sa atong shampoo, para dili siya maka-damage, dili siya too acidic for our scalp, and medicines, so dili maka-irritate ang mga drugs sa atong stomach. The foundation for buffer chemistry began with Claude Louis Berthollet's study of chemical equilibrium in the late 18th century. He studied chemical equilibrium in simple terms: the balance of chemical reactions, meaning dili lang isa ka-direction moadto ang reaction. Sometimes it goes forward and backward until it reaches a point of balance. Berthollet proposed that reactions can reach a dynamic balance. Dynamic as abot kay dili siya frozen, meaning reactions are still happening. Pero ang forward o backward na reaction, they move at the same rate. So it looks stable overall. Think about it like a game of tug of war. Both teams are pulling hard pero equal ra ang force. So walay move. Mao na ang pasabot ni Berthollet, a balance where both sides are active. Let's move on to the mid-19th century, where scientists like Robert Boyle and Wilhelm Oswald observed that weak acid-salt mixtures resisted pH changes. Pero unsa ganitong pH? pH measures how acidic or basic a solution is. What Boyle and Oswald found was if mag-add kag little acid or base sa mixtures, wala kaayo nag-change ang pH. This was the first real sign that certain substances can makastabilize the ion sa pH. That's basically how a buffer works. These early findings hinted at the mechanism behind pH stability.
The discovery of buffer action in the late 1800s to the early 1900s. Let's start in the early 1900s. A scientist named Lawrence Joseph Henderson studied how our blood keeps its pH stable. He discovered a natural system called the carbonic acid bicarbonate system, which prevents our blood from becoming too acidic or too basic. Basically, it works like a built-in balancing act. Carbonic acid provides the acid part, while bicarbonate acts as the base. Mao siya ang reason nga nung our blood stays near pH 7.4 even if we eat acidic food or exercise. For example, when you run and breathe heavily, your blood releases more carbon dioxide, and this buffer system helps keep things balanced. Henderson also introduced the word buffer to describe any system that resists sudden pH changes. This idea became the foundation of modern buffer theory. A few years later, in 1916, Carl Albert Hasselbalch built on Henderson's research. He came up with the Henderson-Hasselbalch equation, which helps scientists calculate the pH of buffer solutions. This formula basically tells us how much acid and base are in a solution and how that balance affects the pH. Here are the definitions of the symbols. pKa is the acid dissociation constant, a measure of how strong or weak an acid is. Muna siya ang gatal at what pH the acid is half dissociated, meaning half is acid and half is base. A negative is the base and HA is the acid. Buffers in the 20th century. The 20th century saw buffers become essential in research and industry. They were used in biochemistry to keep enzymes stable. In medicine, to maintain blood pH, and in chemical labs to control reactions during experiments. Outside the lab, we can also find buffers in everyday products. For example, ang imong shampoo. Nag-contain na sila buffers para ma-match ang imong skin's pH. So, para dili sila makakause of irritation. Goods buffers in the 1960s. In the 1960s, Norman Good and his colleagues created a set of special buffers for biological experiments, known as Goods buffers. They were non-toxic, easy to dissolve, and didn't interfere with living cells. Some examples include HEPAs, MOPs, MAPs, and TES. Thanks to Goods buffers, scientists could finally study enzymes and cells in conditions close to how they naturally exist inside the body. Modern understanding and application. Today, buffers are essential in science and industry. They are almost used in every field. In food production, they help maintain flavor and freshness. In medicine, they're used in drug formulation and in storing blood safely. In environmental science, buffers help control the pH of soil and water, which is vital for farming and aquatic life. In the cosmetic industry, they help keep lotions and creams gentle on our skin. Buffers are vital for safety, stability, and accuracy. The role of buffers in life. Life depends on buffers to regulate internal pH. Inside us, we have the bicarbonate buffer in blood, the phosphate buffer in our cells, and even proteins that can balance pH. Without them, our body chemistry would swing wildly, and even small pH changes can stop enzymes from working properly. So to summarize, from Henderson's early discoveries to modern science, buffers have proven to be one of the simplest yet most powerful systems in chemistry and life. They help our bodies stay balanced, our products stay safe, and our experiments stay accurate.